Hi, it's Mr. Dykstra. In this video we're going to talk about how we can identify and recognize various simple biological molecules. There's four main classes of biological chemicals. First one, carbohydrates or sugars, is primarily used for quick energy in the form of ATP for regular cell tasks. The second one, lipids, are highly nonpolar, and their main role is for long-term energy storage. Proteins complete all the everyday tasks of the cell. And nucleic acids are where the cell stores its necessary information or instructions on how to make things. Let's start by looking at some simple carbohydrates. On this screen we have four isomers. They all have the same chemical formula, C6H12O6. So they have the same ratio of carbons and hydrogens and oxygens, but we can notice that they are somewhat different structurally. In fact, all carbohydrates follow the general pattern that for every carbon you would have twice as many hydrogens and equal number of oxygens. So it would be CH2O. Now, this screen here we've got four. Let's start by looking at fructose in the bottom corner. This one stands out because the fact it actually has a pentagon for its backbone rather than a hexagon. And we also see that it has two side chains, two carbons sticking out the side. So that's the easiest way of identifying fructose and separating it from the rest. Now let's move to the other ones. Glucose typically starts by having a staggered set of hydroxyl groups. Both alpha and beta glucose will have this. With beta glucose, the staggering continues, so we end up getting kind of a Z shape for beta glucose. So the hydroxyl groups start down, up, down, up. Alpha glucose, however, starts the same way, but then it ends up having two hydroxyl groups pointing down. Galactose has the first two groups pointing up, and that's an indication of this sugar that we're looking at is galactose. There's some other simple sugars as well. All three of these ones that we see on the screen, we might notice have that same pentagon ring. Where the differences come in, again, we notice the fructose has these two side chains, so again, it's easily recognized from the other two. Ribose and deoxyribose, the difference is right here. One of them has a hydroxyl group, the other has simply a hydrogen. So we have ribose is the general format, but then deoxyribose, we can think of deoxy as missing an oxygen. And that missing oxygen is coming from this hydroxyl group right here. So if we see that there's an oxygen missing, then we can know very quickly that this is a deoxyribose sugar. If both oxygens are present, then we know it's a ribose, and if we have those side chains, it's a fructose. Moving on, these sugar isomers or monomers can actually be bonded together to form more complex sugars such as disaccharides. So here we have an example of a disaccharide being formed. We have the hydroxyl group from one of the monosaccharides and a hydroxyl group from the other monosaccharide are coming together. One of the oxygens is going to end up being the oxygen link between the two molecules, and the other will pick up the two hydrogens and be lost as a water. So water is always formed. This is why they call it a condensation or dehydration synthesis. When we're building molecules together, they end up removing the water, and water ends up being produced. So what kind of disaccharides are there? Well, there's three I'm going to talk about today. The first one, sucrose. What you'll notice is that we have a fructose here as one of the subunits. You see those two side chains again standing out very easily. So that's a quick indication that it's sucrose. The sound kind of gives it away. Fructose, sucrose. Sounds very similar. The second monomer in the pair is always a glucose. So if we have identified the one as being fructose, then we know by default the other has got to be a glucose. When we look at lactose, what we'll notice pretty quickly here is we've got these two up again. That's an indication that we have a molecule of galactose. When galactose combines with glucose as the second subunit, then we get lactose. Lactose, galactose, seems logical to me. The third one, you notice these two structures are essentially identical. So what we have here is two glucoses bonding together. Notice the staggered pattern to start off the hydroxyl groups. So we have two glucoses forming, and together they form one maltose. As for polysaccharides, they form when we start getting many more subunits of sugar being put together. In this screen right now, we see two examples. One is from animals and humans, and the other is from plants. Both of them are forms of glucose. So we're forming them into a straight chain. In fact, they're both using alpha glucose. And what we'll notice is that these rings or chains can be simplified by a diagram. If I did it like this, represent these rings simply by circles, that all the side chains are pointing up. Let me show you on the side here. These side chains are all pointing up in the same direction. That's a sure indication that this is a starch or glycogen, and that it's going to be used for energy. So alpha glucose, when it's combined in polymers, polysaccharides, is used for energy. The difference between these two is that starch makes these straight chains, so it ends up being like a fiber. So it gives support and structure. So we'd have a whole pile of parallel fibers being formed here that would give strength for wood and so on in the plants. What we notice in the glycogen is we start getting not only the straight chain, but we also start getting side chains off it. And it can branch in many and various different ways, as we see in the diagram here at the bottom. That takes up a lot more space, but it also provides a lot more flexibility. So as humans and animals, we can bend and move and so on without having the fibers get in the way. We also have carbohydrates being used for structure. And again, we have an example here from plants and from animals. So the top here, we have cellulose. This is an example of structural support. It's found in wood, holding up all the trees. What you notice very quickly, if I was to represent these again as circles with the side chains, is that the side chains here are alternating different directions, up, down, up, down. Because of this fact, it won't fit into any of the enzymes that we possess as humans. So we cannot digest anything that has fiber or cellulose in it. And so this is primarily a structural formation. Chitin, found in insects, we notice the same type of pattern. Again, it's going to be a beta glucose, just like cellulose is a beta glucose. So we can represent these by circles again. And the side chains are again alternating. So that's our tip off from looking at the carbon coming off the side. It's almost identical to the cellulose. They're alternating up and down, up and down, up and down. But you might notice something else as well. What you notice here is that there's some nitrogens. They have actually a second side chain, and that second side chain has nitrogen in it. So anytime you see a polysaccharide, a large sugar, it's got a side chain with some nitrogen in it. I don't even have to look any further. If you see nitrogen, boom, it's chitin. You've solved the identification already. And then we know that chitin is used in the exoskeletons of insects, for example.